Okay, thank you, Professor Chairman. Actually, I prepared a presentation for 20 minutes, but I will try to get it done between 10 to 15 minutes. This, I must say, is quite a technical topic, as I've seen through all, mostly monitoring, which is about the topic that we talk about. But I hope that this one will give you a new insight to the tools that how we develop tools to improve water quality monitoring. We know that water quality monitoring, we always got lot of limitations. Hopefully, this sort of device may help you in the future. So what I'm going to talk about is waterborne pathogens. As you see this morning, Goal 3 actually compared waterborne diseases. So if you have a good device to monitor this, it definitely will help to solve the problem. This is particularly important when we have a sanitation problem for 2.4 billion people. If you know sanitation, waterborne diseases will be very rampant. And also 50% of the microbial intestinal infection is basically caused by bacteria, and with cholera as the most important. Alright, the main bacterial diseases transmitted through water, as you can see in this table, basically the Vibrio species, the Salmonella, the Shigella, the E. coli. So Cryptoporidium also happen quite often sometimes. And we focus on Vibrio cholerae, bring about cholera, and E. coli, bring about gastroenteritis to develop our device as an example. The normal technique for determination of this pathogen, I think most of you are familiar, microbiology technique. You culture it, wait for maybe a day, get the colonies, you count, and then you try to analyze them, which is the species involved. This is very simple procedure, no doubt, but it takes time, it takes skill, it has to be done in the lab. And there are more advanced methods now, more sophisticated, where you can do using DNA, nucleic acid, to do hybridization. You multiply the DNA of this species of pathogen using polymerase chain reaction, and then you detect it using the gel electrophoresis and so on. This is a very good method, also latest, but it is a very complicated procedure. It needs a lot of chemicals. Again, another method that can be done in the lab and take time and scale. So human capacity building is important in this sort of method. Just to show you why is it rapid technique is important. Recently, in the analysis of Cryptoporidium pathogen, there are many steps, as you can see, there are 6 steps here, because you not only need to analyze the bacteria itself, you need to analyze also the oocysts. So you have to break the oocysts and release the RNA, and then you do the usual analysis. This is again a very time-consuming method and highly technical. Therefore, there's a company trying to develop what we call a rapid microanalytical system. So all these steps are actually included in one of these devices. So all you do is that you put in your thing, whatever chemical, whatever sample there, and it will do the job in a few hours. This is a great improvement. But we think that this is still need a lot of human intervention. It may not be able to bring to the field for analysis as well. Therefore, we move on. We are trying to explore a technology called biosensor. In the biosensor, basically, it's a device that have a recognition layer, which can be an antibody and enzyme or DNA. They can recognize for example, antibody, antigen. Enzyme is lock and key principle. DNA is hybridization of the complementary. So whatever signal generated during the biochemical process will be transduced by transducer. This can be a current or electrical signal or optical signal. I think I have to skip this. This is how it works. There are many advantages, particularly decentralization laboratory. Costly hardware replacement, no reagent, rapid, remote sensing is possible. If we make such a device, we can put it in the monitoring in lake or whatever, you can remotely translate the data to your whatever device. So the main idea is to convert a big equipment like that into a small handheld device with an electrode. We focus on this organism because they're genetically they are known. Their DNA is already known. We can use the DNA for identification. So basically all you do is you immobilize the probe of the organism onto an electrode, and then you require a redox indicator. This indicator will only respond when there's hybridization with the sample DNA. When there's no hybridization, it just simply bound off the DNA and then you cannot respond. But if the DNA from the pathogen come along, it will hybridize, and then this redox mediator will go into the DNA strand, and then from here you will get a signal. So this signal is a theoretical signal that we are getting. So this is basically the fabrication process. It seems very complicated, but the work is quite easy. So there are two ways of fabricating the biosensors. And this is the response. You can see that the big peak is hybridization. When you have the DNA sample, the E. coli is there, the Vibrio cholera is there, but when it's not there, you don't get any signal or current. So this actually confirms that the sensor is working. And there are a lot of things to do, like optimization of all the parameters to give the best performance. And in fact, the sensor actually is proportional to the concentration of the DNA of the E. coli or the Vibrio cholera. So if you have the sample so much amount, you can detect that based on the current measurements. And how about the selectivity? That's also very important. Okay, so complementary DNA is 100% matching. Okay, so three bases different of the DNA, you get about 46%. If you put the E. coli DNA there, you get only 9%. So the response is also selective because the DNA is selective to the complementary. Another important characteristic of the biosensor is that it must have a long-term storage stability. You don't use it today, tomorrow cannot use. You have to bring to a field. Maybe you go on the field trip of 1 month, 2 months. So at least the sensor that you have should be able to do the job for the next few months. So we tested it. It should be stable up to at least 1 to 2 months. It can be usable if you keep in your fieldwork kit. And this is, we validated the sensor by comparing it with the plate culture method. If you use real water, we found that actually we can detect E. coli, the fecal E. coli, and then the culture method also show there are colonies there. But if you look at tap water and bottled drinking water, we don't detect, the current is very low, we don't detect any DNA. Then you culture this sample, you don't get any colony of the bacteria. Similarly with seawater, we found some DNA and also we culture, we found some E. coli cell as well. So this is validating that the biosensor actually can be used in the field. Similarly with the Vibrio cholerae biosensor, and for the Vibrio cholerae strain, they all positive response. They give you from 50 to about 100% response. But if you put in the DNA of the extract DNA from other species of bacteria, you get very low response, only about 10%. Okay, so again, this shows that actually they can respond to different type of bacteria. I think I skip this. This is just to show you that the plate culture method again. So if you have the sample, have the bacteria, DNA, you have DNA, you have the bacteria. No DNA, no bacteria. So at the end of the day, actually we are moving towards here now. We can develop a reader that can be calibrated with the concentration of DNA, and then we have an electrode that contains the sensor inside. And all we need to do is put a drop of water sample, and then you will know whether your sample contaminated with E. coli or Vibrio cholerae. So this is a battery-operated device, just like a mobile phone. So you can bring it anywhere with the electrode together. They no longer need to bring water sample to a lab and do all the analysis. That's what we plan to do for the future. So as a conclusion, basically the concept is actually workable, as you can see from the data, and that's without the need of large amount of chemical and highly skilled manpower. So they no longer need to train human capacity to do bacterial analysis and so on. The use of such analytical device should assist in rapid water quality monitoring program especially for pathogen organism, and suitable for fast screening. At least if you cannot detect the actual amount, you can screen, okay, whether the bacteria is there or not, or the pathogen is there or not. And because the DNA method, it can actually confirm whether whatever you get is this E. coli, a cholerae, or Cristoporidium. Because it's a platform technology using DNA, I presume any pathogen, you have the DNA sequence, you can use this method. So I'm ending my talk now by showing my appreciation to the UNESCO for inviting me and sponsor my trip here to present this. And I also very much want to thank Professor Minoro Yoneda and also financial support from my Malaysian Ministry of Science and Technology. Thank you very much for your attention.
Thank you very much. I think that is very, very interesting presentation. I like to, okay, Professor Hans is already waiting.
Yes.
Yeah, really nice presentation. Do you have to lyse the cells to get at the DNA?
At the moment, we are using sonication, but I think in the future you can actually use a bit of lysing reagent to break the cell. Sometimes heating also you can break the cell to release the DNA. A very simple procedure.
So in your slide where you showed a drop of water.
Yes, that probably already lies with the reagent. Yeah.
So you have to have some sort of lysis.
Yeah, if you don't want, you can heat up the sample
Okay.
to break the cell.
I think it's a really important contribution, but you raised the issue previously of viable versus non-viable organisms, and that's particularly important in water treatment plants. Can it tell the difference between a viable organism and a non-viable if you're only doing the DNA?
Yes, what you mean is that some is the bacteria is dead or alive, right? So normally dead bacteria, the cell will decompose very fast, the DNA will be broken up very quickly, degraded. So normally what we measure is likely to be the viable one, the live one, the DNA. But there's a chance, like I said, it may be measuring the dead bacteria as well, if it just died, you know. But we have to look at that issue also.
Thank you. I just want to ask, the sensor, can it be adaptable to community level?
Yes, because like I said, you don't need any skill to use them. All you need to know is how to read the reader. The sensor itself is already all prepared. Maybe you need to learn how to a little bit of the sample preparation, lyse the cell or something. I would expect this can be very helpful to community.
Yes. And can it be, is it available at large quantity?
Well, there's one catch here. Actually, in lab, it looks very nice, but if you want large quantity, then we have to manufacture. Then the process has to go to the factory. Then we have to optimize everything. So hopefully, if they're successful, then this sort of device may be available for use in the future. But you have to sort out the problem of fabrication first.
Okay.
One question.
Yeah.
Yeah, do you think that in the future it will be possible also to use this method for online monitoring?
For what?
For online monitoring. Will it be possible in the future?
Migratory?
Online monitoring.
Online. Yes, what is electrochemical? Online, we have to learn how to regenerate the sensor because it's a DNA. Once it's bind already, you have to break the binding again for the next sample. So online, at the moment, I think it may not be that easy.
Okay, so thank you, Professor.